Scientists identify a novel strategy to fight viral infections and cancer in animal model, a potential therapeutic strategy to treat viral infection and boost immunity against cancer, as reported in the May 30 online issue of the journal Cell. The work, conducted by scientists at Wake Forest School of Medicine, found that boosting the body's production of type 1 interferon helped clear viral infection and increased immunity against cancer by identifying a sensor involved in suppressing interferon production in an animal model. Interferons are a group of signaling proteins made and released by host cells in response to the presence of several viruses. In a typical scenario, a virus-infected cell will release interferons, causing nearby cells to heighten their antiviral defenses. Interferons also help the immune system fight cancer and may slow the growth of cancer cells. The principal investigator of the study was Wei-Quan Lin, Ph.D., professor of cancer biology at Wake Forest School of Medicine, part of Wake Forest Baptist Health. In the study, Lin's team discovered that RIG-I-like receptors (RLR) mediated interferon (IFN) production, which played a pivotal role in elevating host immunity for viral clearance and cancer immune surveillance. They reported that glycolysis, the first step in breaking down glucose to extract energy for cellular metabolism, was inactivated during RLR activity. That inactivation served as the key to turn on type 1 IFN production. Using pharmacological and genetic approaches, the scientists showed that lactate reduction by lactate dehydrogenase A (LDHA) inactivation heightened type 1 IFN production to protect from viral infection in mice. The study established a critical role of glycolysis-derived lactate in limiting RLR signaling and identified MAVs as a direct sensor of lactate, which functions to connect energy metabolism and innate immunity, Lin said. Type 1 interferons (IFNs), produced by almost all type of cells, played a vital role in host defense against viral infection and cancer immunosurveillance, Lin said. Lin's team plans to conduct additional studies in other animal models in preparation for potential clinical trials. This work was supported by startups and the Endowment Fund of Anderson Discovery Professor for Cancer Research from Wake Forest School of Medicine, National Institutes of Health (NIH) grants R01CA182424 and R01CA193813, and NIH grants R01CA194094 and R01CA197178. The research team also acknowledges the support by the National Key Research and Development Program 2017-YFA05039000, National Natural Science Foundation of China 31,671,413, and Beijing NOVA Program Z161100049161147, and Major Program of National Natural Science Foundation of China 81,790,252. How the immune system keeps the Epstein-Barr virus in check. A protein called PD-1, which is found on immune cells called CD8+ T cells, plays a key role in controlling infection with the Epstein-Barr virus, according to a study published May 30 in the open-access journal PLOS Pathogens by Christian Munz of the University of Zurich, and colleagues. The results from this study indicate that monitoring PD-1 signaling during future vaccination and immunotherapy studies may inform patient outcomes. Since its discovery as a tumor virus by Epstein and colleagues in 1964, the Epstein-Barr virus has been implicated in many serious diseases. This virus is one of the most ubiquitous human pathogens in the world, persistently infecting more than 90% of the adult human population. It drives some of the strongest human CD8+ T cell responses, which can be observed during the initial symptomatic, or primary, stage of infection known as infectious mononucleosis. Despite high viral loads and prolonged CD8+ T cell stimulation during infectious mononucleosis, the Epstein-Barr virus enters latency and is under lifelong immune control in most individuals that experience this disease. Currently, in vivo studies are lacking to understand the comprehensive immune control of the Epstein-Barr virus in most healthy virus carriers, and, in particular, the characteristics of the CD8+ T cells involved in this process. To address this gap in knowledge, Munz and colleagues examined CD8+ T cells in patients with active infectious mononucleosis, as well as in a well-characterized mouse model for Epstein-Barr virus infection. The researchers found that even though CD8 plus T cells express multiple receptors that normally inhibit immune responses, including PD-1, during primary Epstein-Barr virus infection, these T cells appear to retain the ability to produce cytokines, kill infected cells, and proliferate. Importantly, blocking the PD-1 pathway leads to specific defects in controlling the Epstein-Barr virus and increases virus-induced tumor formation in infected animals, indicating that this pathway is important for viral control. This is in contrast to previous studies showing that PD-1 helps keep the body's immune responses in check, and releasing this inhibitory block is important for reinvigorating immune responses against cancer. Because PD-1 function is required to keep the Epstein-Barr virus in check, this study provides evidence against blocking inhibitory pathways in disease settings that require improved immune control of chronic virus infections. Although immune checkpoint blockade constitutes a major breakthrough in cancer therapy, our results suggest that reactivation of persistent virus infections should be monitored as a possible side effect at least during anti-PD-1 treatment, says Bithi Chatterjee, first author of the study. Brush your teeth, postpone Alzheimer's. You don't only avoid holes in your teeth by keeping good oral hygiene. Researchers at the University of Bergen have discovered a clear connection between gum disease and Alzheimer's disease. The researchers have determined that gum disease, gingivitis, plays a decisive role in whether a person develops Alzheimer's or not. We discovered DNA-based proof that the bacteria causing gingivitis can move from the mouth to the brain, says researcher Peter Meidel at Brogelmans Research Laboratory, Department of Clinical Science, University of Bergen, UIB. The bacteria produces a protein that destroys nerve cells in the brain, which in turn leads to loss of memory and ultimately, Alzheimer's. Brush your teeth for better memory. Meidel points out that the bacteria is not causing Alzheimer's alone, but the presence of these bacteria raise the risk for developing the disease substantially and are also implicated in a more rapid progression of the disease. However, the good news is that this study shows that there are some things you can do yourself to slow down Alzheimer's. Brush your teeth and use floss. Meidel adds that it is important, if you have established gingivitis and have Alzheimer's in your family, to go to your dentist regularly and clean your teeth properly. New medicine being developed. Researchers have previously discovered that the bacteria causing gingivitis can move from the mouth to the brain, where the harmful enzymes they excrete can destroy the nerve cells in the brain. Now, for the first time, Meidel has DNA evidence for this process from human brains. Meidel and his colleagues examined 53 persons with Alzheimer's and discovered the enzyme in 96% of the cases. According to Meidel, this knowledge gives researchers a possible new approach for attacking Alzheimer's disease. We have managed to develop a drug that blocks the harmful enzymes from the bacteria, postponing the development of Alzheimer's. We are planning to test this drug later this year, says Peter Meidel. Research confirms gut-brain connection in autism. People with autism often suffer from gut problems, but nobody has known why. Researchers have now discovered the same gene mutations, found both in the brain and the gut, could be the cause. The discovery confirms a gut-brain nervous system link in autism, opening a new direction in the search for potential treatments that could ease behavioral issues associated with autism by targeting the gut. Chief Investigator Associate Professor Elisa Hill-Yarden, RMIT University, said scientists trying to understand autism have long been looking in the brain, but the links with the gut nervous system have only been recently explored. We know the brain and gut share many of the same neurons, and now for the first time we've confirmed that they also share autism-related gene mutations, Hill-Yarden said. Up to 90% of people with autism suffer from gut issues, which can have a significant impact on daily life for them and their families. Our findings suggest these gastrointestinal problems may stem from the same mutations in genes that are responsible for brain and behavioral issues in autism. It's a whole new way of thinking about it, for clinicians, families and researchers, and it broadens our horizons in the search for treatments to improve the quality of life for people with autism. The autism gene and the gut-brain link. The study reveals a gene mutation that affects neuron communication in the brain, and was the first identified as a cause of autism, also causes dysfunction in the gut. The research brings together new results from preclinical animal studies with previously unpublished clinical work from a landmark 2003 study led by Swedish researchers and a French geneticist. The study of two brothers with autism by Professor Christopher Gillberg, University of Gothenburg, Professor Maria Rostam, Lund University, and Professor Thomas Borgeron, Pasteur Institute, was the first to identify a specific gene mutation as a cause of the neurodevelopmental disorder. This mutation affects communication by altering the Velcro between neurons that keeps them in close contact. While the 2003 study was focused on identifying the genetic basis for autism, Gillberg and Rostam also took detailed clinical notes of the brothers' significant gastrointestinal problems. Researchers in the gut-brain axis team at RMIT have built on this clinical work with a series of studies on the function and structure of the gut in mice that have the same Velcro gene mutation. The team found this mutation affects gut contractions, the number of neurons in the small intestine, the speed that food moves through the small intestine, responses to a critical neurotransmitter important in autism, well known in the brain but not previously identified to play any major role in the gut. Collaborator Associate Professor Ashley Franks, La Trobe University, also found significant differences in the gut microbes of mice with the mutation and those without it, even though both groups were kept in identical environments. While this specific Velcro mutation is rare, it is one of more than 150 autism-related gene mutations that alter neuronal connections, Hill-Yardin said. The link we've confirmed suggests a broader mechanism, indicating that the mutations that affect connections between neurons could be behind the gut problems in many patients. New research horizons on the gut-brain axis. Hill-Yardin, an ARC Future Fellow and Vice-Chancellor's Senior Research Fellow in the School of Health and Biomedical Sciences at RMIT, said the work identifies a new target for the development of therapies specifically designed to work on neurotransmitters in the gut. We've also identified that there's a need to better understand how existing autism medications that target neurotransmitters in the brain are affecting the gut, she said. Another promising path for future research is investigating how gene mutations in the nervous system relate with microbes in the gut. We know these microbes interact with the brain via the gut-brain axis, so could tweaking them improve mood and behavior? While this wouldn't reverse the gene mutation, we might be able to tone down its effects, and make a real difference in the quality of life for people with autism and their families. The research, with collaborators from University of Gothenburg and Lund University, Sweden, Baylor College of Medicine, US, University of Minho, Portugal, La Trobe University, the University of Melbourne, Florey Institute of Neuroscience and Mental Health and Monash University, Australia, is published in a forthcoming edition of Autism Research. The work was supported with funding through an Idea Development Award from the United States Department of Defense, DoD, Congressionally Directed Medical Research Programs, CDMRP, Autism Research Program, the Victorian Government through the Operational Infrastructure Scheme, the National Health and Medical Research Council, NHMRC, and the Royal Melbourne Hospital Neuroscience Foundation.